السلام عليكم أعزائي الطلبة. في المحاضرة السابقة تكلمنا حول موضوع antigen processing. Now we will talk about the cell-to-cell interaction and immune responses. How the immune cells collaborate in eliminating the antigens. There will be a collaboration between the T cells and B cells. And what's the role of T helper cells in regulation of the immune response? So, the teaching objective of this lecture is to discuss the central role of T helper cells in immune responses, describe the cell-to-cell interaction which occur in antibody response to T-dependent antigen, generation of cytotoxic T cells, and activation of macrophage and natural killer cells. To discuss the response to T-independent antigen, to discuss the mechanism of killing by cytotoxic T lymphocyte and macrophages. Now, what is the central role of T helper cells in immune responses? After T helper cells recognize specific antigen presenting cells, they can initiate several key responses. These include selection of appropriate effector mechanism. Whether the selection will be for B cell activation or cytotoxic T cells activation and generation. After that, there will be an induction of proliferation of appropriate effector cells. After selection process, there will be a proliferation of either B cells or cytotoxic T lymphocyte. Then there will be an enhancement of functional activities of other cells like granulocyte, macrophage, and natural killer cells, as it will be seen in the figure below. This figure will illustrate how the antigen presenting cells present antigen to T helper cells. Depend on which type of antigen present in antigen presenting cells, the T helper cells will be differentiated and it will stimulate either B cells activation and proliferation of B cells to produce antibodies, or the T helper cells will activate cytotoxic T cells, and these cells in turn it will be proliferated. The next step in these two cells is the induction of activation of certain types of cells depend on the antigen presented by antigen presenting cells, and there will be activation of certain types of cells by certain molecules secreted by the activated T cells. Examples, the antibody will stimulate antibody dependent T cells response, and like killer cells, sometimes there will be activation of natural killer cells, activation of macrophage, even activation of eosinophils to produce different types of cytokines to eliminate the target antigens. There are 4 types of subpopulation of T helper cells, and this is the naive T cells, or we call the T helper 0, T helper 1, T helper 2, and T helper 70s. When naive T helper cells encounter an antigen in secondary lymphoid tissue, they are capable of differentiation into either inflammatory T helper 1 cells or helper T helper 2 cells, or pathogenic or regulator T helper 17 cells. How it will be differentiated, which type of T helper cells will be differentiated according to the cytokines present in the environment in relation to the antigen. So, which are distinguished by the cytokines they produce. Whether the T helper 0 cells become T helper 1, T helper 2, or T helper 17s depend upon the cytokines in the environment, which is influenced by the antigen presented by the antigen presenting cells. For example, some antigens stimulate interleukin 4 production, which favor the generation of T helper 2 cells, while other antigens stimulate T helper 12 production, which favor the generation of T helper 1 response. The T helper 1, T helper 2, and T helper 17 cells affect different cells and influence the type of immune response, as it will be shown later on. So, cytokine produced by T helper 1 cells will activate macrophage, and it will participate in the generation of cytotoxic T cells, resulting in a cell-mediated immune response. In contrast, cytokine produced by T helper cells, it will help to activate B cells, resulting in antibody production. In addition to T helper 2 cytokines also activate a granulocyte, while T helper 17 cells, which is designed as such by their production of interleukin 17s, differentiate in a human in response to interleukin 1, interleukin 6, interleukin 23. Interleukin 17 will enhance the severity of some autoimmune disease, including multiple sclerosis, inflammatory bowel disease, and rheumatoid arthritis. So it is a regulator T cells. This figure shows the different types of T helper cells and how these cells will be differentiated according to the microenvironments around these type of cells. So the T helper cells will be converted to T helper 1, and it will mediate cytotoxic T cells or cell mediated immunity, or it will be differentiated into T helper 2 cells that will regulate B cells response and antibody production by their microenvironment, or it will be converted into the pathological T helper 70s that will mediate different types of autoimmune disease. So, T helper 1 cells will activate the macrophage, and this will in turn enhance phagocytosis and activation of other cells by different types of cytokine produced by the macrophage, or the T helper cells will activate certain types of granulocytes like eosinophils or mast cells. Also, it can activate B cells to produce different types of antibodies, and the net result is the elimination of the target antigen. Activation of certain T helper cells, it will downregulate or inhibit the proliferation by the other cells. What is the meaning of this? If the antigens will select the T helper 2 population, there will be a regulatory mechanism that will inhibit the proliferation of T helper 1 and vice versa. So, equally important, each subpopulation can exert inhibitory influence on the other. Gamma interferon produced by T helper 1 cells inhibit the proliferation of T helper 2 cells and differentiation of T helper 17 cells. And the interleukin 10 produced by T helper 2 cells inhibit the production of gamma interferon by the T helper 1 cells. In addition, although not shown, interleukin 4 inhibit the production of T helper 1 and differentiation of T helper 17. Thus, the immune response is directed to the type of response that is required to deal with the pathogen encountered. Cell-mediated immune response for intracellular pathogen or antibody response for the extracellular pathogen. Now, cell-to-cell interaction and antibody response to exogenous T-dependent antigen. Here, the role of T helper 2 in the regulation of the immune response and selection of B cells to produce antigens and coagulation of T cells in this process. A hapten carrier model. What is the hapten? You should refer to the previous lectures to learn more about this type of carrier model. Historically, one of the major findings in immunology was that both T cells and B cells were required for antibody production to a complex protein combination of B and T cells. A major contribution to our understanding of this process come from studies on the formation of the anti-hapten antibodies. Studies with hapten carrier conjugate established that first, T helper 2 cells recognized the carrier determinant and B cells recognized haptogenic determinant. Second, interaction between the hapten-specific B cells and the carrier-specific T helper cells was the self-MHC restricted. And third, B cells can function both in antigen recognition and antigen presentation. وهذا تكلمنا عليها في المحاضرات السابقة how we consider the B cells as antigen presenting cells. Now, what's the role of B cells in regulation of the immune response and killing of the certain types of extracellular antigens? B cells occupy a unique position in immune response because they express the immunoglobulin on their surface and the class II MHC molecule. So they therefore are capable of producing antibody having the same specificity as that expressed by their immunoglobulin receptor. In addition, they can function as an antigen presenting cells. So they carry both functions. So these figures show an attempt of a hapten carrier conjugate model. The mechanism is thought to be the following. The hapten is recognized by the immunoglobulin receptor on the B cells. The hapten carrier is brought into the B cells by endocytosis. Then the antigen hapten molecules will be processed and peptide fragment of the carrier protein are presented to helper T cells. Activation of T cells result in the production of cytokines that enable the hapten-specific B cells to become activated to produce a soluble anti-hapten antibody, and it will be shown in the figure later. So this figure shows after activation of the T cells, cytokine bind to certain molecules on B cells that will stimulate their proliferation and differentiation of these type of B cells that produce specific antibodies to hapten courier molecules. So there are multiple signals delivered to B cells in this model of T helper 2 cells B cell activation and interaction between the B and T cells. Binding of antigen to the immunoglobulin receptor deliver one signal to the B cells, but that is insufficient. A second signal delivered by the costimulatory molecules is required. The most important of these is the CD40 ligand on the T cell surface that bind to the CD40 on the B cells to initiate a delivery of a second signal. So there will be a stimulatory molecule and co-stimulatory molecules, both of them required for the activation of B cells. Now, cell-to-cell interaction in the primary antibody response. B cells are not the best antigen-presenting cells, as we said in previous lectures. In primary immune response, dendritic cells and macrophage are the most efficient antigen-presenting cells. Nevertheless, with some minor modification, the hapten carrier model of cell-to-cell interaction described above also applies to interaction in the primary antibody response. So, in a primary immune response, the T helper cells first encounter antigen presented by dendritic cells or macrophage. The prime T helper 2 cells can then interact with B cells that have encountered antigen and are presenting antigen peptide in association with MHC class II molecules. Here, the B cells require two signals for their activation. One signal is derived from the binding of antigen to the surface immunoglobulin, and the second signal is by binding of CD40 ligand during the T helper 2 B cell to cell interaction. In addition, cytokines produced by T helper 2 cells help B cells to proliferate and differentiate into antigen secreting plasma cells. Now, cell-to-cell interaction in a secondary immune response. As a consequence of a primary response, many memory T and B cells are produced. Memory B cells have high affinity immunoglobulin receptor. This is due to affinity maturation, which allow them to bind and present antigen at a much lower concentration than the required for the macrophage and dendritic cells. So we call more specific antigen presenting cells. In addition, memory T cells are more easily activated than the naive T cells. Thus, B T helper cell interaction are sufficient to generate a secondary immune response, mostly primary immune response. It is not necessary to prime memory T helper cells with antigen presenting by dendritic cells or macrophage. Now we will talk about the immunoglobulin class switching and what is the role of cytokines in class switching. Cytokines produced by the activated T helper 2 cells not only stimulate proliferation and differentiation of B cells, but they also help to regulate a class switching for antibody production. So different cytokines influence the switch to different classes of antibody with different effort function. In this way, the antibody response is tailored to suit the pathogen encountered. The best example is the immunoglobulin E antibody class switching to parasite infection. Cell-to-cell interactions and antibody response to exogenous T-independent antigen. This type of cells will not depend on T helper cells. The antibody response to T-independent antigen do not require cell-to-cell interaction. The polymeric nature of these antigens allow for cross-linking of antigen receptor on B cells. And this cross-linking of antigen will result in the activation of B cells. So, no secondary response, affinity maturation, or class switching occurs in this type of antigen. Responses to T-independent antigen is due to activation of subpopulation of B cells called CD5 positive B cells, also called B1 cells, which distinguish them from the conventional B cells that are CD5 negative, or we call the B2 cells. So what is the importance of B1 cells, or we call CD5 positive cells? These cells are the first B cells to appear on ontogeny. They express only surface IgM molecule without IgD molecules on their surface. They produce primary IgM antibodies from a minimally somatically mutated germ cell lines. Antibodies produced by these cells are of low affinity and often polyreactive. It is not a specific type 1. Most of the IgM in the serum is derived from the B1 cells. The CD5 positive or B1 cells do not give rise to memory cells. An important characteristic of these cells is that they are self-renewing. Unlike conventional B cells, which must be replaced from the bone marrow, B1 cells are found in the peripheral tissue and are the predominant B cells in the peritoneal cavity. B1 cells are the major defense against many bacterial pathogens that are characteristically have a polysaccharide in nature. The importance of these cells in immunity is illustrated by the fact that many individuals with T cell deficiency or defect are still able to resist many bacterial pathogens. So this type of B cells carry a vital role in eliminating many types of bacterial pathogens. Now, the cell-to-cell interaction in cell-mediated immune response. Generation of cytotoxic T lymphocyte in response to exogenous antigen in the cytosol. Cytotoxic T lymphocytes are not fully mature when they exit from the thymus. They have a functional TCR that are recognized antigen, but they are not able to lyse the target cells. They must be differentiated into a functional effector cytotoxic T cells. Cytotoxic T cells differentiated from the pre-CTL cells in response to two signals مثل ما شفنا بالبي cells activation. Specific antigen in contact with the MHC class 1 molecule on the stimulator cells and the cytokines produced by the T helper cells. especially the interleukin 2 and gamma interferons. So there will be a stimulator and co-stimulator for their activation of this type of T lymph. So this figure shows how the activation of T cells by the antigen binding to specific MHC class 1 molecules and their cytokines that produce within the microenvironment. Now, what is the features of cytotoxic T lymphocyte mediated lysis? CTL killing is antigen specific. To be killed by the cytotoxic T lymphocyte, the target cell must bear the same class 1 MHC associated antigen that are triggered pre-cytotoxic T lymphocyte differentiation. The cytotoxic lymphocyte killing require cell-to-cell direct contact, and this we call the kiss of death. The cytotoxic T lymphocyte are triggered to kill when they recognize the target antigen associated with the cell surface MHC molecule, usually type 1. Adjacent cells lacking the appropriate target MHC molecules are not affected. Cytotoxic T lymphocytes are not injured when they lyse the target cells. لذلك يعني كل واحدة تبوسها تقتلها. Therefore, each cytotoxic lymphocyte is capable of killing sequentially numerous target cells. How the cytotoxic T cells kill the antigen presenting on MHC class 1 molecules. Cytotoxic T cells utilize several mechanisms to kill the target cells, some of which require a direct cell-to-cell contact, and others that result from the production of certain cytokines. In all cases, death of the target cells is a result of apoptotic cell death. So, one of the mechanisms of cytotoxic T killing is the by fast anti-necrosis factor mediated killing. Once the enriched cytotoxic T lymphocytes express fast ligand on their surface, which bind to the fast receptor on the target cells. In addition, tumor necrosis factor alpha secreted by the cytotoxic T lymphocyte can bind to the tumor necrosis receptor on the target cells. So we have both the fast ligand and tumor necrosis factor and tumor necrosis factor alpha, both binding, then are mediate killing of these type of cells. So the fast and tumor necrosis factor receptor are a closely related family of receptors, which when they encounter their ligand form a trimer of the receptors. These receptors also contain the death domain in the cytoplasmic portion of the receptor, which after trimerization can activate caspase enzymes that induce apoptosis on the target cells.
In our previous video lectures we understood that CD8 plus T cells become cytotoxic T cells. Cytotoxic T cells kill the infected cells. By doing so they eliminate the reservoir of infection. These cells are also known as killer T cells or cytotoxic T lymphocytes, abbreviated as CTLs. They are named so because they directly kill the cells that display foreign antigens on their surface. The main function of cytotoxic T cells is to kill cells that have been infected by viruses and bacteria. They are also involved in killing of tumor cells and transplanted foreign cells. Let's see how these cytotoxic T cells work. When there is an intracellular infection, for example viral infection, dendritic cells from the site of infection migrate to nearby lymph node and present the viral antigens in the form of MHC1 antigen complex. Naive CD8 positive T cells recognize the bound antigen. At the same time, nearby activated T helper type 1 cells are secreting interleukin-2. Recognition of the antigen by CD8 positive T cell, and the signals from T helper cells, result in the activation of CD8 positive T cell. This T cell now proliferates and differentiates into effector cytotoxic T cells and memory T cells. These effector cytotoxic T cells secrete more interleukin-2 and become self-stimulating. Now, these cytotoxic T cells leave the lymph node and migrate to the site of infection, to attack virally infected cells. We know that all nucleated cells express MHC1 molecules on their surface, which under normal conditions, display the host's proteins or self-antigens. But when there is an infection, these cells display foreign antigens. Therefore, in this case, viral antigens are displayed on the infected cell surface as MHC1 antigen complex. The effector cytotoxic T cells recognize and bind these MHC1 antigen complex on the infected cells. The cells which are killed by cytotoxic T cells are known as target cells. To perform its killing function, cytotoxic T cells need to make a direct contact with the target cells. This direct contact is mediated by T cell receptor and coreceptor CD8. T cell receptor binds the antigen displayed by MHC1 molecule, and CD8 binds the MHC1 molecule. Once a contact has been established with the target cell, cytotoxic T cells now undergo degranulation. In other words, the granules of the cytotoxic T cells migrate to the site of contact, and granule content is released. Cytotoxic T cells contain specific cytotoxic granules which contain preformed cytotoxic effector proteins such as perforin, granzymes, and granulysin. These proteins are synthesized and loaded into the granules as soon as the naive CD8 positive T cell encounters its specific antigen. Perforin forms pores in the membrane of the target cell. This damages the target cell and it also forms a passage for other granule contents into the target cell. Granzymes enter the target cell through these pores and cause apoptosis or programmed cell death. Thus, the target cell is degraded from within. Granzymes trigger apoptosis in the target cell by activating enzymes such as nucleases, and by damaging the mitochondria. Granulysin also has antimicrobial activity and it contributes to apoptosis of the target cell. These degraded cells are rapidly ingested and digested by phagocytes. The cytotoxic T cells moves to another infected cell to perform the killing function. Besides their main function as killer cells, cytotoxic T cells also secrete cytokines. The major cytokines secreted by killer T cells are interferon gamma and tumor necrosis factor alpha. The main role of these cytokines is to activate macrophages and recruit them to the site of infection.
Now, what is the role of the macrophage? Cell-to-cell interaction and cell-mediated immune response activation of the macrophage in response to endogenous antigen in the vesicles. Macrophages play a central role in the immune system. They are involved in the initial defense against pathogen as part of the innate immune system. Second, antigen presentation to the T helper cells. And the third, virus carry various effector functions. Example, cytokine production, bactericidal, anti-homicidal activities. Indeed, macrophages play an important role not only in the immunity but also in reorganization of the tissue. However, because of their potent activities, macrophage can also damage to the tissue, so it may harm the host cells. Many of these macrophage functions can only be performed by the activated macrophage. So what is the activated macrophage? Macrophage activation can be defined as a quantitative alteration in the expression of various gene products that enable the activated macrophage to perform some functions that cannot be performed by the resting macrophage. الصور يعني إذا resting macrophage carry their function, it will destroy the host cells. So there should be a tight control for their activities, and this is regulated by the different aspects of the immune system and their gene activation. Macrophage activation is an important function of the T helper 1 cytokine, which is mediated by the T helper 1 cells. When T helper 1 cells get activated by the antigen presenting cells such as the macrophage, they release gamma interferon, which is one of the two signals required to activate macrophage. Lipopolysaccharide from bacteria or tuberculosis alpha produced by the macrophage exposed to bacterial product deliver their second signals. So there will be also the first signal activation and the second signal activation. As I discussed in the previous lecture on the innate immune response, effector mechanism employed by the macrophage include first the production of alpha interferon, alpha tumor necrosis factor, which can induce apoptotic cell death. Second, nitric oxide and other reactive oxygen nitrogen intermediates. The third is the reactive oxygen intermediates, and the fourth, cationic proteins and hydrolytic enzymes. And these all products mediate killing by the macrophages. Macrophage activation by the T helper 1 cells is very important in the protection against many different pathogens and very important pathogens. For example, pneumocystis carini in AIDS patient, and it is an extracellular pathogen, is controlled in normal individual by activated macrophage. It is, however, a common cause of death in AIDS patient because they are deficient in T helper cells. Similarly, Mycobacterium tuberculosis is an intracellular pathogen that resides within the vesicles of the infected cells. It is not efficiently killed by the macrophage unless they are activated. Hence, this infection is a problem in the AIDS patients. Thank you for listening.